All right, we're now going to talk about our last section in chapter 2 on proteins. And with the function of proteins, proteins serve as catalysts. Enzymes are proteins in metabolic reactions. They act in defense, they aid in transport, they contribute to structural support, they cause movement, they perform regulation, and they provide storage. The general protein structure is one or more strands of amino acid monomers. There are 20 different amino acids found in living organisms. Each has an amine and a carboxyl functioning group, both covalently linked to the same carbon atom. Carbon also covalently bonded to hydrogen, and different side chain structures form R groups, which distinguish different amino acids from one another. General protein structure, amino acids are covalently linked by peptide bonds between them. They're formed during dehydration synthesis, so an anabolic reaction, between an amine group of one amino acid and the carboxylic group of another. Hydrogen is lost from the amine group, and then hydroxide is lost from the carboxylic acid of another amino acid. The N-terminal end has a free amine group, and the C-terminal end has a free carboxyl group. So we have here with amino acid, we have the amine and the carboxylic acid, and then we have our group here, noting that one of 20 different structures here for the amino acid. Dipeptide here showing two different, basically two peptides that form that dipeptide bond. And then we have polymer protein, which is a long strand of peptide bonds, or any variations on how that shape would actually be, but it should say a significant number of peptide bonds. And then we'll talk more about that in a second. General protein structure, strands of amino acids, an oligopeptide, then talking about different polypeptides, have between 3 and 20 amino acids, a polypeptide between 21 and 199 amino acids, and a protein if it's more than 200 amino acids. Glycoproteins are proteins with carbohydrate attached. Glycoproteins are on erythrocytes, red blood cells, determining A, B, or O blood groups. So what are the monomers of proteins? They are amino acids, and the name of the bond between them is a peptide. What are the names of structures that contain two amino acids, as opposed to, so basically a dipeptide, 3 to 20 amino acids, oligopeptide, 21 to 199 amino acids, polypeptide, and 200 or more amino acids, protein. What general term is used to refer to any of these structures, or proteins, except the structure composed of two amino acids, which is a dipeptide. So you should then now be able to, for the next and final section, name the categories of amino acids, distinguish between nonpolar, polar, and charged amino acids, and give examples of amino acids with special characteristics, as well as describe the different types of intramolecular or attractions that participate in both the folding of a protein and in maintaining its three-dimensional shape. Distinguish between the four structural hierarchy levels of proteins, and explain what is meant by denaturation, and list factors that can cause it. So the categories of amino acids, they're organized into groups based on their R group. There are non-polar amino acids, contain the R groups with hydrogen or hydrocarbons. They're grouped with other non-polar amino acids by hydrophobic interactions. Polar amino acids contain R groups with other elements besides hydrogen or hydrocarbons. They form interactions with other polar amino acids and with water. The categories of amino acids, they're organized into groups based on their R group. Right, one of those 20 of those amino acids. There are charged amino acids, which contain the R groups with a negative or positive charge. They form ionic bonds between negatively and positively charged amino acids, and they're hydrophilic. They don't like water, or they love water, excuse me. Amino acids with special functions. Proline can cause a bend in the protein chain. Cysteine can form a disulfide bond. And methionine is the first, or methionine, some will say, it's the first amino during protein synthesis. And this one's kind of hard to see. I don't think I'm going to try to go over this too much. This is a little difficult. I thought it was going to be more simple to see, but it's a bit of a strain trying to pull these out here. So let's see if we can get these a little closer. There we go. So we have the nonpolar with glycine, alanine, valine, isoleucine, leucine, phenylalanine, and tryptophan. And we have our polar with serine, threonine, asparagine, and asparagine, and glutamine and tyrosine. Charged, we have glutamate, aspartate, histidine, lysine, and arginine, or arginine, some would say. Proline, cysteine, and methionine are the special functions whereas like the proline will allow the bend of the protein chain, and the cysteine will form that disulfide bond, and methionine will always be the first amino acid as a protein sequence, but it can be removed following synthesis of the protein. As far as amino acid sequence and protein conformation, primary structure is a linear sequence of amino acids. So we have our peptide bond showing the amino acid, and basically it's a little closer look at that amino acid and that peptide bond. So the conformation is three-dimensional shape of the protein. That's crucial for protein function. Levels of organization beyond the primary structure, basically in describing the conformation. Arrangements that are dependent upon the intramolecular interactions between amino acids, and they're obtained through folding with the help of specialized proteins, chaperones. Intramolecular interactions. Hydrophobic interactions with non-polar amino acids are farther from water. Hydrogen bonds between polar R groups between amine and carboxylic acid groups. Ionic bonds between negative and positive R groups. And disulfide bonds between cysteine amino acids. So we have our secondary structures, basically where the patterns that just may repeat actually several times. They confer some unique characteristics. There are two types. We have the alpha helix, like the one shown here. It's a spiral coil. It's elasticity to fibrous, showing elasticity to fibrous proteins like skin and hair. And the beta sheet here, it's basically a planar pleat type arrangement, and there's flexibility to globular proteins, an example being enzymes. Okay. Tertiary structure, the three-dimensional shape of the polypeptide chain. There are two categories, the globular protein and the fibrous proteins. The globular proteins fold into a compact shape, and the fibrous proteins are extended linear molecules. Quaternary structure is present in proteins with two or more polypeptide chains, and a very simple example is hemoglobin with its 4 polypeptide chains. Prosthetic groups are non-protein structures that are covalently bonded to protein, like lipid heme group and hemoglobin protein. Denaturation is a conformational change to a protein, disturbs protein activity. It's usually irreversible, basically where the protein actually basically configuration actually unwinds, basically due to increased temperature or response to changes in pH. And once it unwinds, it's going to basically die. It's not going to be usable. It needs to maintain its structure in order to actually be usable for the purpose that it's created. Other causes of denaturation: pH changes, interferes with electrostatic interactions, and some other intermolecular bonds. If there's a change in blood pH, can be lethal. Yeah, I mean, that's the whole thing. Lethal to the cell, or lethal to the protein itself, I should say. And if significant enough, lethal, the more proteins that are going to die, eventually is going to lead to obviously affecting the organ, and the organ system, and the organism. So what distinguishes the tertiary and quaternary levels of organization of a protein? You have your globular and your fibrous proteins and tertiary. So we're basically talking about 3 polypeptide bonds. And the quaternary levels, we have 2 polypeptide bonds or more. And basically with the quaternary levels, we refer to something like hemoglobin, or hemoglobin is probably the best example of the quaternary. What happens to a protein when it denatures? It basically unwinds due to a change in the pH or a change in temperature. Basically, when that protein denatures, when it's exposed to higher than normal hydrogen concentrations, it changes its structure and basically loses its functional capabilities, basically dies. And again, the more that proteins die, proteins affect everything, right? Affect all form of cells. So consequently, if it happens significantly enough, that could affect the entire organism itself. And that is chapter 2.